So, welcome again to Biotech Scientific Masterclass series, where researchers from around the world present their latest results and findings to a global audience. So, my name is Michael Erklens, and I'm part of the research and development team of Biotech. Today, I'm happy to introduce this masterclass series, which should provide a unique platform for scientists to share their work, connect with others in their field, and receive feedback from experts and peers. So after the talk today, there will be time for the interactive question and answer session. So during your talk, you will be able to use the chat function to comment aspects of the talk or ask questions. You can directly place a question for the Q&A session by changing the chat mode to the Q&A mode. So you can see on the bottom right, you can see a little question mark, so you can use the question mode for placing your question for later. So our speaker today is Dr. Samar Damiati. She's associate professor at the University of Sharjah in the United Arab Emirates. So Professor Damiati received her PhD degree in 2013 in the Department of Nanobiotechnology at the University of Natural Resources and Life Science, BOKU, in Vienna. Later in 2019, she joined the KTH Royal Institute of Technology in Stockholm, Sweden, as an invited researcher. Since 2014, she was first assistant professor, but became later associate professor in the Department of Biochemistry at the KAU. Basically, Damiati's research is focused on bottom-up synthetic biology to construct artificial cell membranes that mimics the feature and behavior of biological membranes. So currently, she's using microfluidic technology to construct artificial cell models and drug delivery systems. And her talk today will be about this topic. With this, I would like to finally hand over to Professor Damiati and her exciting talk. So thank you and see you later for the Q&A session.
So hello everyone. First of all, I would like to thank the organizers for giving me the opportunity to present my work today and to share with you my research about synthetic biology and how to build artificial membranes from scratch. First of all, let me start my presentation with a simple introduction about synthetic biology. In synthetic biology, we combine science and engineering in order to design and build a new system that mimics the natural one. One application of synthetic biology is building artificial cells. I mean here to build whole cells or part of the cell, such as the membrane. Synthetic biology is divided into two main approaches to build artificial cells: top-down and bottom-up. Although these two approaches are different, they are complementary to each other. The top-down approach is based on living organisms and eliminating non-essential cellular components. Then it can be exploited as a host cell that engineers new function. In contrast, bottom-up approach is based on single molecular assemblies. This strategy constructs a biomimetic cell by assembling living and non-living molecules that are built on biological and synthetic system. In my research, I focus on bottom-up approach. We know about the complex nature of biological membranes. We know it's a challenging task to investigate the structure and function of natural membranes. For example, it's not easy to investigate membrane proteins. We know that more than 60% of drugs on market target membrane proteins. It's important to study structure and functions of membrane proteins for medical and pharmaceutical research. But we don't have enough information about these proteins because it's difficult to synthesize and to purify membrane proteins. Also, they can be easily denatured once they are isolated from the lipid environment. Therefore, we need to build artificial lipid membranes that can be used for membrane analysis. I mean here to investigate their structure and functions. So here we come to our question. Can we rebuild the cell membrane? The simple answer is yes. We can build artificial cell membrane in a simpler form using basic units, using simple materials such as nucleic acids, amino acids, and lipids. Still, it's not easy and requires many trials to construct functional system. So here, to build a model of the cell membrane, we need first to build the lipid membrane, and here it's possible to build monolayer or bilayer. Many model membranes have been already developed. The most common biomimetic models used for studying membranes structure and processes, as well as for investigating the membrane activity, are lipid membranes, lipid monolayers, lipid vesicles, and supported lipid bilayers. Each of these systems has advantages and disadvantages, but they all mimic the lipid arrangement of natural cell membrane. Let's focus on the cushion-supported lipid membrane. It's possible to form the lipid bilayer membrane in direct contact with the cell surface, or adding an intermediate layer that separates the lipid membrane from the solid surface. Intermediate layer or spacer molecule here can be used to support the lipid membrane and to provide a space. So once we have the space here, we can insert large membrane proteins. Also, we need this intermediate layer to reduce the risk of protein denaturation because it minimizes the direct contact between the solid surface and the protein. In this study, we used surface layer proteins. S-layer protein is, we used here this S-layer protein as a spacer molecule. S-layer protein is a bacterial protein, can be crystallized in suspension on lipid membranes and also on many solid substrates such as gold and silicon dioxide. So, to monitor the formation of our artificial membrane, here we used quartz crystal microbalance with dissipation monitoring. So, QCMD is a real-time technique that measures frequency and dissipation. Frequency is related to the mass absorbed on the sensor surface, and energy dissipation is related to the viscoelastic properties. So, to build the S-layer supported lipid membrane, in the first step, S-layer protein injected into the QCMD cell. As you see here, we have a change in the frequency because here the mass increased onto the gold sensor. Then, to prepare the lipid membrane, liposomes were injected and mass significantly increased. Here we have a significant changes in frequency and dissipation. So, mass here first increased and then decreased. The decrease here because of the liposomes fusion. So here the trapped water molecules are released and bilayer structure, lipid bilayer structure are formed to the S lattice. So the changes here in frequency and dissipation signals indicated the formation of a good lipid membrane. So we can use it for the next step, which is to reconstitute membrane protein. So the second step, in building artificial membrane model based or focused mainly on the protein. And here we are trying or aiming to use the simple molecules, as we mentioned before, building from scratch. So we will collect simple materials. So here we worked with voltage-dependent anion channel, VDAC, VDAC protein is, we find that the VDAC protein in the mitochondrial outer membrane. It's a channel protein, regulates the passage of small molecules and ions. Also, we have here, we used a special strategy to synthesize VDAC protein. So we use the cell-free protein expression system. So it's the cell-free protein expression system is a simple tool for small-scale protein synthesis that overcomes the limitations of cell-based system and can synthesize the protein in a short time, and also it doesn't require cell culture. So simply here we need two basic components, the genetic materials such as messenger RNA or DNA. So these materials encoding the target protein, and a reaction solution containing all materials that are needed for transcription and translation. So here, 3 steps are involved in the second part to build artificial membrane. First, we designed DNA primers. Then we use these primers to synthesize VDAC protein using the cell-free protein expression system. And finally, protein molecules reconstituted into our artificial model, the S-layer supported lipid membrane. So here is a summary of these steps. We designed different DNA primers. Then we mixed the primer with the cell extract, and we confirmed the VDAC protein synthesis by Western blot. Also in this study, we developed 2 strategies for membrane protein insertion into the S-layer supported lipid membrane using the cell-free protein expression system. And the first strategy, we used pure VDAC proteins. So here we synthesized the protein following the protocol for expression system, the cell-free expression system. Then after purification, we started to inject the VDAC protein into the QCMD cells. Changes here in frequency and dissipation are not significant. You see here small changes, and that doesn't confirm protein insertion. So a complementary technique is needed. So we combined QCMD with electrochemical impedance spectroscopy to measure membrane resistance. We measured their resistance before and after insertion of VDAC protein. During the membrane formation, membrane resistance increased for each additional layer on the gold sensor because each layer acts as a barrier to charged molecules. Then, after VDAC reconstitution, membrane resistance significantly decreased as a result of protein reconstitution and channel formation. So as you see here, increasing the protein concentration, when we add the protein at different concentration. So here we increased the number of channels that reconstituted into the S-layer supported lipid membrane. So more ions crossed the membrane, and therefore we have less resistance. This is the first strategy. In the second strategy, we synthesized VDAC protein directly in the presence of the S-layer supported lipid membrane. So simply here, we mixed VDAC DNA with the cell extract with the reaction mixture here and inject the solution directly into the QCMD cells. After incubation, we collected the sample for the Western blot analysis, which confirmed the protein synthesis. However, injection of the mixture caused significant changes in frequency and dissipation. Also, the membrane resistance significantly decreased. We assume here there is a strong interaction between the cell extract and the lipid membrane, which may cause partial lysis and deorganization of the membrane architecture, as the cell-free solution can be considered a highly crowded and detergent-like environment. Anyway, we showed a successful construction of one artificial membrane, which is the S-layer supported lipid membrane with active channels, active VDAC protein, using simple materials. Now I would like to show you another artificial model that has been also developed in our lab, the lipid vesicles. Liposomes, emulsions, nanodiscs are common examples on lipid vesicles. We can prepare these particles as nanoparticles or microparticles. In our lab, we prepared nanodiscs. These nanodiscs mimic the high-density lipoprotein particles, and they are promising carriers for drug delivery, and also we can use the nanodisc to investigate water-soluble proteins and water-insoluble protein. Nanodiscs can be produced based on the self-assembly of phospholipids and membrane scaffold protein. So, as I mentioned, it's possible to use the nanodisc with water-soluble proteins and water-insoluble protein. So if we work with water-soluble proteins, the protein in this case can be absorbed into the nanodisc surface. But if we have water-insoluble protein, the protein molecules can be inserted into the lipid membrane. In this study, we worked with bovine serum albumin, which is water-soluble protein that attached to the nanodisc surface. Fast protein liquid chromatography is used to purify and characterize the generated nanodisc. Here are the FPLC chromatograms for the nanodisc with and without albumin. Nanodisc with albumin have extra peaks, elution peaks, and also the main peak is wider. So, from these results, we also confirmed that the generated nanodiscs are highly stable. We measured here the in vitro release. The nanodisc without albumin didn't show an initial bulk release of membrane scaffolding protein, which indicates slow degradation. But here for the nanodisc with albumin, it shows a faster release kinetics because we have here membrane scaffold protein and also albumin. Anyway, we produced here another successful artificial membrane model that can be used for different analysis, for different types of analysis. So to conclude, designing artificial cell membrane models improves our understanding of cellular life. Supported lipid membrane is a promising platform enabling investigation of membrane protein in an environment close to nature under controlled conditions. Our study employed a promising strategy for membrane protein based on bottom-up approach, starting from a DNA sequence and ending with a functional channel reconstituted into a biomimetic environment. Synthetic high-density lipoprotein like phospholipid nanodiscs present a promising carriers for drug delivery and for membrane protein investigation. Finally, I would like to thank my collaborators, my colleagues, my lab mates, and of course, I would like to thank you all for your attention. Thank you.
Thanks a lot, Professor Damiati, for this exciting talk. So you can now put your camera on so that everyone can see you.
Yes, hello.
Ah, nice. So, hi, nice to meet you. So, yeah, synthetic biology is a really emerging field in the last years, and it's really fascinating. So, your talk gave us nice insights in these the used technologies. So I would say we can directly start with the question and answer session. So, and I have, of course, the first question. So you tried the cell-free expression of membrane proteins. So, and I think last year and the year before, there were some papers about using synthetic polymers for the cell-free expression and experiments with these proteins. And it seems to be much more stable than MSPs. Did you ever try synthetic polymers?
With this system, no. We mainly depends here. We depends mainly on lipid particles, not the polymeric particles.
Okay, because you used the MSPs for the cell-free expression and then the integration in the lipid membrane. So, okay.
If you are talking about the nanodisk, so here we have phospholipids and the membrane scaffolding proteins, not polymer not polymer molecules.
Okay, because then you maybe can also still have the proteins in their native lipid environment and maybe can insert them in the membrane. But I'm not sure if this was ever tried before.
I don't know.
Okay, so let's come to the question and answer session. So I will, where is it? There, the question and answer mode. So the first question is from Abdullah Al-Muteri. So which genes have you examined?
We used here VDAC genes. We designed different genes isolated from the bacteria, and then we tried using the cloning system, the normal cloning system, just to design different primers, because not all genes or not all sequence can synthesize protein. If we use the cell-free system, it's easy with the cell-based system. But for the cell-free, we need here to design carefully the sequence, keeping in mind or keep a space or extra nucleotides between the Shine-Dalgano sequence and the ribosomal binding site. So the short answer here, we have VDAC genes from bacterial sources.
Great, thank you. So the next question was asked by Thorsten Bremer. Which protein yields were you able to achieve with the nanodisk, and does excess MSP interfere with downstream application?
Okay, with the nanodisk, we used albumin. We simply order it, so it's already pure and ready to use. And for any excess molecules, it can be isolated by the fast protein liquid chromatography. So excess of proteins, excess of lipids, by the process of PLC. Okay, I hope that answers your question.
So Esme Duchet, I hope it's pronounced correctly, Duchet, is asking the question, are S-layer proteins a common spacer, or do they have specific advantages to other spacers?
Okay, it's possible here to use polymers. It's common to use polymers, not S-layer proteins in particular. Also, it's possible to use tether molecules, which is simpler. But here it depends on which molecules do it like to, or which molecules do you have in your lab. So, but here for the S-layer, because mainly it depends on the self-assembly process, and we can repeat the experiments many times on the gold sensor. We didn't face any problem here with the S-layer proteins. That's why we prefer to use the S-layer as a spacer molecule. For the advantages, it's only because it's a reproducible system.
Thank you very much. So Tina Hartmann is asking, you said at the end that nanodiscs offer a promising opportunity for drug delivery. Have you been able to gain any further insights about this?
We focused on albumin, so we worked with the protein investigation. Especially if you check the publications, most of the people work with protein, insoluble proteins. So insoluble proteins can be easily integrated into the lipid membrane. But here because we have limited number of studies focused on water-soluble proteins such as albumin, we prefer to work with something new with limited information. So we focused here on the protein, not drug, but still, it's possible to use the nanodisk as drug carriers.
Thank you. And the next question is from Abdullah Al-Muturi again. Have you exposed the artificial cell membrane to different environments?
I'm not sure if I understand this question correctly. If you mean if it's possible to insert different proteins into our model, yes, we tried with 2 proteins and 1 peptide. So it was possible here to insert grammacidin, which is a peptide, small peptide, and VDAC. For hemolysin, it's another protein, but it doesn't work for the layer-supported membrane. We assume here because of the shape or the structure of the protein, VDAC has a cylindrical shape. So with a cylindrical shape protein, we can reconstitute it into this layer-supported lipid membrane. So also here, these 2 proteins have the same molecular weight, but mainly here, it depends on the structure and the shape of the protein to be inserted functionally and from the active channels into the artificial model.
Okay, thank you. I think the question was maybe also related to buffer conditions or salt, ionic strength, and so on. But I'm sure you also tried different conditions, right? So for the place.
Yeah, we optimize the conditions. Yeah.
Okay, great. Thank you. So the next question is from Abdelazi El Gamous. So how can we target cancer cells by the nanodisk technology?
It's similar to the high density lipoprotein, so it's easy to have it all over to distribute it in the blood. So how it can be targeted, you can functionalize it actually, but still it's easy to do. The distribution process here is easy in the body. It's similar to, or it mimics the HDL.
So with functionalization, you mean, for example, antibodies or something like this, right? So
yes.
Okay. So, but did you try the functionalization with antibodies, or are you still at the step before? So the creation of this kind of materials.
Not with the nanodisk.
Okay. Thank you. So Abdelaziz Tlili asking the question, primers used to amplify which DNA, human or animal?
Human DNA. Because we work with this VDAC, it's human protein in outer membrane of mitochondria.
So Ahmed Javed is asking, I was unable to understand the purpose of the gold surface. So was its purpose to the detection of the voltage or current outputs during the experiments? Are there other examples of substances we can use as surface besides gold?
Okay, here we have the solid-supported strategy. So we need this the substrate of gold. And why it's gold, not silicon dioxide, for example, because as you mentioned, it's important for measuring the electrochemical measurements. So we need here, when we apply the current, when we measure the volt, we need this kind of metals such as gold. For the other substances, you need to modify. For example, for silicon dioxide, you need to modify it first, then you can use it for electrochemical measurements. So it's easier here to use gold.
Thank you. So then maybe Derek, a question from my side. So because of the gold, so can you also combine your use technique with, for example, SIRS spectroscopy to check or to measure the placement of your protein on the lipid environment, or is the S protein layer too big, too thick?
Yeah, you can use different techniques. We already did it with the QCMD with surface plasmon resonance SPR. So many techniques can be involved.
Okay, so the next question is from Sebastian Igounet. So he's asking, in the S-layer system, are you able to influence protein orientation in the membrane?
Here it's based on spontaneous insertion and so you need to have active channel in its open state, so you can that enables the ions movement. We know sometimes if it for the biomimetics we have freestanding system and salt supported system. For active channels, some proteins prefer the freestanding system, and it cannot be reconstituted into salt supported system. So, but for VDAG, it works well for the salt supported system. For hemolysis, for example, it prefers freestanding system, not the solid system. But for about the orientation here, it depends on self-assembly process.
Mm-hmm. Okay, so we're running out of time. So maybe a last question. So, what are the further steps that can be taken? So, do you have any plans?
Okay, here we need to because we are using the self-expression system, we need to ensure which sequence can be used to synthesize the protein. So this is one step, find the right sequence and designing different sequences. And for the S-layer supported membrane, we need to investigate more proteins. So just to know if it's a good membrane for a library of proteins or limited number of proteins.
Mm-hmm. Okay, thank you. So, first of all, thank you very much for your exciting talk and your time today. So, it was really, really informative. So, with this, I would like to also give you information about the next masterclass. So, let me share the there. Can you see the screen?
No.
Nice. Okay, so the next masterclass is held on the 13th of September, so same time as every time, and is held by Dr. Kirill Natetstin from the Columbia University. And his topic will be about structural mechanisms of TRPM7 activation and inhibition. So I hope we will see you again